Hello everyone, welcome science fans. It's the end of another long teaching day, but I'm still going to knock out another one of these videos just to get us started on a new playlist, a new series of videos on respiration. So hopefully if you're following an A-level syllabus just like ours, you'll have finished photosynthesis, so you'll know a little bit about things like electron transport chains, the ATP synthase molecule, which all come back in this topic. Okay, so respiration divides up neatly into 4 stages: glycolysis, the link reaction, the Krebs cycle, and the electron transport chain, with a few other side reactions bits at the end. So we're going to go straight into it with session number 1, which is glycolysis. Okay, the structure of the mitochondria and how glycolysis breaks down glucose into a thing called pyruvate. Okay, a little reminder of hopefully some stuff that you should have done already. So glycolysis, being the first stage of both anaerobic and aerobic respiration, occurs outside the mitochondria. The rest of this material ends up inside the mitochondria. So start with this question, which is actually more of a sensible question than it looks initially. How many ways is respiration like combustion? Okay, so it's always a good idea in this topic to think back to your GCSE understanding of respiration. Okay, respiration being glucose plus oxygen, right, making carbon dioxide and water. Okay, so if you want to take a little moment now to stop and think of as many ways as you can that respiration is like combustion, we're then going to look at some of the differences. Okay, so if you've done that, well, they're both burning reactions effectively. They are both taking a fuel, a carbon-rich fuel. In the case of respiration, that's glucose. In the case of combustion, it's any carbon-based, generally fuel, wood, coal, gases, fossil fuels, anything like that, and burning them, okay, and releasing the energy from within them using oxygen. Okay, so it uses oxygen to release the energy. Okay, a lot of that energy comes as heat, and that's the same with respiration and in combustion. However, there are some key differences. Okay, respiration has a much lower activation energy, okay, because it's enzyme controlled. That's what enzymes do. They lower activation energy. Combustion, you've really got to start that getting going, and that's quite good because I've got a big pile of logs in my house, and I'm pretty glad they don't start combusting spontaneously when I'm not in the house. Okay, so higher activation energy to get those going. Respiration, again, this is another big advantage, respiration over combustion. It releases the energy from its fuel in really nice, small, steady steps. Okay, combustion is fast, it's all at once, so it will consume the oxygen as quickly as it can, and obviously the more oxygen is provided, the faster the reaction will go. Right, that will be no good inside you. Your cells need energy in nice, neat little packets. Respiration is slightly more efficient, still pretty inefficient. About 75% of the energy made from breaking down glucose ends up as heat, and 25% ends up in the bonds of ATP. It's a rough idea, depends on tissue. But in combustion, obviously the vast majority is heat. There's a little bit of light, a little bit of sound maybe, but the vast majority is heat. And that obviously can be very dangerous in you. Okay, so respiration is a good way to release energy in a cell, and combustion is a good way to release energy in your log fire. So here we can represent that graphically. The idea of respiration is that we turn our fuel and oxygen into water and carbon dioxide. Remember, the same ingredients, but we turn it into those products in gradual steps, releasing the energy slowly and using that energy to generate ATP, adenosine triphosphate. Whereas combustion, you get the reaction going and then whoosh, all your energy is released at once. Okay, so as a reminder, shouldn't need it, but from GCSE, it's always good in this topic to come back to the GCSE understanding, so you know where the glucose has gone in, where the oxygen has come in, where the carbon dioxide has come out, and where the water has come out. Because that arrow, these ingredients turn into these products that you learned at GCSE, hides a hell of a lot of detail. And if you've done the photosynthesis topic, you will know all about that already. Okay, so let's start with where, the locations. Now, you will have learned at GCSE and through AS level probably that respiration occurs in the mitochondria. Mitochondria do respiration, and that is predominantly true. However, it's the very first step we're talking about today doesn't actually happen in the mitochondria. It happens in the cytoplasm. Okay, happens in the cytoplasm. So glycolysis takes place in the cytoplasm. and leads to this thing called pyruvate, this substance called pyruvate, and that is what enters the mitochondria. And that's an examiner's favourite trick, is to try and catch you out by going, okay, we've got an isolated sample of mitochondria, and we're going to do an experiment on it. We give it some of its fuel, we give it some of its respiratory substrate. What should we give it? They want you to go, oh, you give it glucose, don't you, because it's respiration. No. Without the cytoplasm there, you give pyruvate. Okay? Quick recap. Again, you should learn this at the beginning of the AS level, so you should know this already. The structure of the mitochondria. You've got two membranes, the outer membrane and the inner one. The inner one is highly folded into these cristae. That increases the surface area because just like in photosynthesis, one stage of this, the last stage, electron transport chain, involves membrane-bound carriers that are embedded in that membrane. So that's very important for separating the two environments, the intermembrane space from the matrix, because we're going to build a proton gradient. Right? We're going to use this energy to build a proton gradient, just the same as happened in photosynthesis. Okay? The matrix there, as we said, is the goo in the middle, which contains ribosomes and its own circular DNA, because this did used to be a free-living organism back in evolutionary history. Mitochondria and prehistoric ancient relatives of the eukaryotic cells entered this symbiosis that's been the sort of seed for evolution of all eukaryotes ever since. Okay, and it's an incredibly successful thing, but they've still got a legacy, a history of being free-living prokaryotic-like organisms in terms of their circular DNA, their own ribosomes, meaning they can make and manufacture their own RNA and their own proteins. Okay, and of course all those proteins are involved in respiration, pretty much, because that's what the mitochondria does. So here is glycolysis, and it is literally just a load of chemical reactions. Now, if you look at the syllabus, you just have to know what is going on. So let's go through this nice and slowly to start with. So you start with glucose. Glucose is a 6-carbon molecule, and as such is very, very stable. Okay, it's a really stable molecule. Now, to get it to react, to get it to do anything, right, we need to actually give it some energy to get it going. And again, it's good that sugars are a stable molecule because we don't want them spontaneously breaking down and releasing their energy sat in a bag in our cupboards. Otherwise, your baking box would effectively be a highly effective bomb, right? So we need glucose to be stable, right, which is good. But then we want to start this process. You've got to put some energy in to get this thing going. So remember we said it's got a lower activation energy, not no activation energy. So 2 ATP go in to start with, right? And as you can see, the thing that comes out is 2 ADP. Not 2 ADP and PI, because what the PI are actually being used to do is phosphorylate the glucose. Okay, so we phosphorylate the glucose once, doesn't destabilize it enough. We phosphorylate the glucose again. Double phosphorylation leads to an unstable enough molecule, this glucose phosphate stuff. Okay, and that will then break down into triose phosphate. Now you might be thinking, triose phosphate, where have I heard that before? And if you've done the Calvin cycle in photosynthesis, you'll know that that's the thing that comes out of the Calvin cycle and then gets built back up into glucose. Right? So this is literally the reverse of that process, of breaking the glucose back down. Triose phosphate, incredibly important. Okay? And particularly for on the OCR syllabus, they make a massive deal of the overlap between respiration and photosynthesis. Okay? Now, from this point onwards, see that splitting arrow there, you've got to think about everything doubled. Now, a lot of the diagrams will go per triose phosphate, right, or per glucose. And you've got to know which, because as you can see here, we've got to think about everything in terms of doubles, right? Triose phosphate then gets turned into pyruvate, okay? That happens by an oxidation reaction. So this stuff called NAD, right? That one there is a nicotiniamide adenine dinucleotide, I think it stands for. We don't need to know that, right? That stuff there gets reduced, so nicks a load of electrons and a proton, right? It's called a coenzyme, okay? Just like NADH in photosynthesis, it nicks a proton and a load of electrons, becomes reduced, and triose phosphate is oxidized, okay? That releases energy, okay? And that energy does a thing called substrate level phosphorylation. Okay, substrate level phosphorylation is using an enzyme substrate reaction, using the energy within a substrate, in this case triose phosphate, to directly phosphorylate ADP to ATP. Now this is different from photophosphorylation that you will have learned about if you did photosynthesis, and it's different from oxidative phosphorylation, right, which we're going to talk about later in the electron transport chain. This is directly using a substrate, and it's called substrate level phosphorylation. Now the reason I say you have to know that there's two, right, two triose phosphates go to two pyruvates, is that each of those triose phosphates turning into pyruvate has the energy to phosphorylate 2 ADP to ATP. So in total, when we've doubled this up, we end up with 4 ADP turning to 4 ATP. Each one also has the energy to reduce 1 NAD to reduced NAD, or you can say NADH. Okay, that's the same thing. They're equivalent. Okay, so you've got to know, if a question's asking you, you've got to get straight in your head, are you being asked how many NAD per glucose in glycolysis or per triose phosphate? Okay, so overall, right, we've got two triose phosphates generating 4 ATP in total. Those 4 ATP come out. We've already put 2 in, so in total, you're 2 ATP up. Okay, you're 2 ATP up. So that's the first point in respiration where you get ATP. Now, we're going to do anaerobic respiration much later in the topic, okay? But this anaerobic respiration, that's where the ATP production stops, okay? So where you've learned previously that anaerobic respiration produces much less energy than aerobic, that's why. Because another sort of word for anaerobic respiration pretty much is just glycolysis, okay? Now that pyruvate, we're going to talk about the aerobic route. It gets used in the next stages. If it doesn't, it'll sit around, break down to lactic acid, but more on that later, okay? So your glycolysis gets double phosphorylated using the phosphates and the energy from the 2 ATPs. You get then glucose phosphate, which breaks down to 2 triose phosphates. Those triose phosphates are oxidized to 2 pyruvates, generating 4 ATP and 2 reduced NAD. So you're 2 ATP up and you're 2 reduced NAD up. Now those reduced NAD or NADHs are going to carry energy. They're going to carry reducing power over to the electron transport chain, and they're going to be used a little bit later. Okay, so there's our labels. So if you're making a diagram of this, you can pause there and jot down some diagrams. Glucose is phosphorylated to make it more reactive. Phosphorylated glucose splits into 2 3-carbon triose phosphates. Energy released from the oxidation of triose phosphate to pyruvate is used to form 4 ATPs. That is substrate level phosphorylation, and hydrogen, protons, are given off and used to reduce 2 NAD to make 2 reduced NAD. Now those of you who are sophisticated chemists might be thinking, hang on a minute, adding protons isn't reduction. I know reduction is gain of electrons. The electrons go too. Okay, you do get, it is reduction, NAD is reduced, but also a proton is added. Those two things happen at the same time. So it might look a bit funny if you're a chemistry student, but both ways of describing this are correct. Okay, so this is actually really good evidence for evolution. The fact it happens in the cytoplasm, and the fact there are other organisms way, way, way back down the evolutionary tree that that do this, show us we all have a common ancestor. So this could be a question. How is glycolysis? So glycolysis is evidence for evolution. Why? And the answer would be, well, it's the same in all organisms. This process is the same regardless of your archaea, prokaryote, eukaryote, doesn't matter what it is, it's the same in all organisms, suggesting a common ancestor. And in 2016, actually, there was a eukaryote discovered that doesn't do respiration in the same way. But the glycolysis reactions, the first stages, are still the same. Okay? Right. So, common question, which is basically where we're going to end this. What are the products of glycolysis? Well, you get your reduced NAD, 1 per triose phosphate or 2 per glucose. You get your ATP, you've put 2 in, you get 4 out total per glucose, right? So a net gain of 2 per glucose. You get 2 lots of pyruvate, and that pyruvate still got a lot of those carbon-carbon bonds. So that is then actively transported into the mitochondria to enter the link reaction and the Krebs cycle, which are coming next. And I will see you then. Bye-bye.